right, so today we are going to finish up the cell size and membrane structure notes that we started taking while I was still in school. So what I want you guys to do really quick is I'd love for you to split screen your screen so that you can actually finish taking these notes so that you have them in addition to watching the video because I am not going to be posting the teacher version. You would probably want to have this stuff filled out. Okay, so I know you guys have spent the last couple of days looking a lot at the cell membrane, at its structure, at its function, but I just want to make sure that we go over these things really quickly as well. Okay, so we already know this. All cells are surrounded by a thin and flexible barrier that we call the cell membrane or the plasma membrane, and this membrane has two major functions. The first function is to provide a barrier that really is there to separate the inside and the outside of the cell. Okay, so providing a barrier between the inside and outside of a cell. Okay, so if we don't have that barrier, we don't have a cell. The second thing that this membrane does, in addition to creating that primary structure of the cell, is it helps transport materials or control what materials are transported in the first place into, and out of the cell. So it controls what's able to enter and leave that cell. Okay, so our cell membrane has a lot of smaller parts. It's going to be composed of some lipids. Okay, the two primary lipid types are going to be cholesterol and then our phospholipids. We'll have proteins that are embedded in there. Those are the two major things that you've seen so far and that we'll talk about in class. And then we also have some carbohydrates that are embedded in this membrane. Okay, so if you guys have been reading your book, you'll have seen that our cell membrane is called the fluid mosaic model. And what I want you to think about as you're looking at this image, it's static, but all of these materials are constantly moving around, okay? So here we have our outside of our cell, our extracellular portion, filled with a bunch of watery fluids, and then we have our internal portion of the cell, which is going to be our cytoplasm, again, a watery fluid. Okay, so the main thing that we have, or the main component of our cell membrane, is the phospholipid. And you guys have looked a lot at phospholipid structure these past couple of days. We can see that it is arranged in two layers with those hydrophilic heads on the outside and the hydrophobic tails on the inside, okay? We call those two layers our lipid bilayer. Okay, and that's going to be the primary component of our cell membrane. Now, within that lipid bilayer, you guys do see these like ring-like structures. That is our cholesterol. Remember, it is a steroid lipid. And it's going to help maintain our membrane structure if the temperature is too hot or too cold. So essentially, if the membrane gets a little too warm, it keeps it from being too fluid. If it gets too cold, it keeps it from being too rigid. We want like an ideal, perfect amount of fluidity. Okay. We also have our proteins. So there are a lot of different types of proteins that are embedded in the membrane. You have protein channels, you have surface proteins, but these proteins are really there to help transport charged, and I'm going to add in here polar molecules as well, in and out of the cell. Okay. And then finally, this you haven't really seen too much yet. Each of our cells is actually able to communicate with the other cells. And the way it does that is through a carbohydrate kind of chain. It just kind of helps those cells identify themselves. Okay. So again, I said the major structural component is our phospholipid. Remember, the head of our phospholipid has the phosphate group. So this is going to be the polar or hydrophilic portion. Okay, it's composed of a phosphate functional group, which we know is charged or polar, and it also has a molecule of glycerol. Okay, and then the tails, we have two of them. We have two fatty acid hydrophobic nonpolar tails. So I'm going to say nonpolar or hydrophobic tails. Okay, these are composed of two fatty acids. You guys saw in your earlier activities that those fatty acids can be saturated and kind of more of a straight chain, or they can be unsaturated, which will give it that double bond and that bent chain. Okay. Now, the reason that we have that bilayer is because our phospholipids arrange themselves into the two layers to shield those hydrophobic tails from those watery environments inside and out of the cell. So again, those hydrophobic tails, hydrophobic means water-fearing. They don't want to be near that watery solution, so they are shielding themselves from the water on either side of the cell. So again, that's the water in the cytoplasm or the water in the extracellular solution. So again, it's a flexible structure, forms a barrier between the cell and its surroundings, and it's filled with proteins and cholesterol and a whole bunch of things, okay? Now, that's our overall structure. Our overall function is to kind of control what is entering and leaving our cell. So we should know, does the cell let every single thing in or out? No. It lets only some things in, and it doesn't let other things through. So we say that our membrane is semi-permeable, or another way of saying that is selectively permeable, meaning it is able to select or determine what can pass or what cannot pass. So selective and semi-permeable, same thing. It just means that some substances can pass directly through. While others cannot. Okay, so kind of looking at this image here, we see a perfect description or depiction of semi-permeability. Here we see that these blue molecules are able to cross that phospholipid bilayer directly, and these orange molecules, they can't. Okay, so here we're seeing that our blue molecules can pass, but the orange molecules cannot pass. And in yesterday's activity, you looked at some of the properties that determine what can pass and what cannot pass. And I'm just going to go over them now. So things that can pass through the membrane and can pass through the membrane easily are going to be small nonpolar molecules. So yesterday you guys saw this as things like it could be nitrogen gas, but more often we talk about like oxygen gas or carbon dioxide. So these are really tiny small nonpolar molecules like gases. They're going to be able to pass really, really easily. Another example of a group that can pass really easily will be larger nonpolar substances. So polarity does have is a big factor determining what can pass and what can't. So if you were to look at some smaller lipids, like some of our steroid lipids, like a cholesterol molecule or something like that, or testosterone or estrogen, those things would be able to pass just because they're nonpolar. And remember, we have this whole nonpolar section in the middle here. It's like a nonpolar sandwich, and the things that are able to pass are the things that can interact really well with that nonpolar section. Now, things that can pass by, so these can pass really easily. Things that can pass by but have a lot of difficulty doing so would be like some small polar molecules. So I like to think about an example of this as being like water. It can pass, but it passes too slowly to actually support life. So I'm going to say that really small amounts of these can kind of squeeze through here and there, but not too much. Okay, typically they need a little bit of help. Now, molecules that can definitely never pass through, okay, is going to be anything charged. So our ions, they're just repelled by that nonpolar middle of our phospholipid bilayer. So our ions, they cannot pass through without the help of a transport protein, okay? And then most polar molecules, like an amino acid, or a sugar molecule, or even water needs help, right? Those things can't pass through fast enough to support life. So these are going to be the types of things they would require the help of a transport protein. Or I'm going to call it a protein channel, and we'll talk more about those in a little bit. Okay, so thinking about why our membrane is selective in the first place, like why is this an important thing? Why does it control what can leave and what cannot? This is because it's really important to be able to control this so that our cells can maintain homeostasis. Remember, homeostasis is just an organism or a cell's ability to maintain that stable internal environment even though their external environment is changing, okay? So our cell membrane is going to maintain homeostasis by only letting in the necessary substances that we need, like oxygen, right? Or glucose we need for cellular respiration so that we can make energy that fuels our cells, or proteins that we may need to build the new proteins that our body needs, as well as other nutrients. Okay, so our cell is going to let in certain amounts of things through the membrane, either directly through or using transport proteins to get that stuff that we need into our cells. And then remember, our cell also needs to be able to remove substances that are waste products, okay, or just things in general. So I'll say to remove substances like CO2, or you guys saw urea as an example, which is produced, it's uric acid. It's what you take excrete in your urine, things like that are waste products, right? So I'm going to say CO2, urea, examples of waste that build up in our cells, or it could be products that we're making within our cells. Maybe we're making lipids that need to be sent elsewhere. Maybe it's our liver excreting glycogen, right? And that's going to be broken down by our cells to make glucose. Hormones, again, that would be a big example. They are long-distance signaling molecules. So when your body produces estrogen or testosterone or cortisol, it needs to send it throughout the body. Or even things like neurotransmitters. Okay, those are going to be the things that tell your cells to actually communicate with our with your brain. So all of those things are things we need to remove from our cell, and our cell membrane determines how much of that gets removed and when. Okay, so for the rest of the day, you guys are going to be working on an assignment. I will see you soon, and I hope you have a good one.